Before starting this webinar, we have a disclaimer to read. This educational session is not intended for the purpose of providing medical advice. All information, content, and materials is for informational purposes only and are not intended to serve as a substitute for the consultation, diagnosis, and/or medical treatment of a qualified physician or healthcare professional. The information supplied through webinars or on our webpage or by any representative or agent of the MSA Coalition, whether by telephone, email, letter, or some other form of communication, is for informational purposes only and does not constitute medical, legal, or other professional advice. So good day to everybody. Today is the Multiple System Atrophy's third webinar, From Bench to Bedside, the Promise and Hope of MSA Research. This series will highlight researchers who have applied for and won a grant from the Coalition's Seed Grant Program. That program began in 2013, has resulted in 56 MSA-focused research projects for a total of $2.85 million. Grant proposals addressing any of 4 distinct disease categories: pathogenesis, diagnostic markers, preclinical, and clinical, are submitted each year. The proposals go through a rigorous review process by MSA researchers who volunteer their time to support this work. The finalists are then approved by the Coalition's Scientific Advisory Board and ultimately by the MSA Coalition Board of Directors. Everyone in this process is dedicated to finding a treatment or cure for this disease. We all have skin in the game. Let's introduce everyone who will be a part of this webinar today before starting the webinar. We'll start with the researcher whose work will be the focus of today's webinar. Vic Khurana is Chief of the Division of Movement Disorders at Brigham and Women's Hospital and Harvard Medical School. He is principal faculty at the Harvard Stem Cell Institute, an associate member of the Broad Institute of Harvard and MIT, and a NYSCF Robertson Stem Cell Investigator. His clinical and research interests relate to neurodegenerative diseases with a focus on Parkinson's disease and related dementia, and rare disorders, including multiple system atrophy and ataxias. Dr. Khurana is a medical graduate of the University of Sydney and obtained his PhD in neurobiology from Harvard University as a Fulbright scholar. He completed his neurology residency and fellowship at Brigham and Women's and Massachusetts General Hospitals, alongside postdoctoral training with Susan Lindquist and Rudolf Janisch at the Whitehead Institute for Biomedical Research at MIT. He led early studies to identify and reverse pathologies in human stem cells derived from Parkinson's patients. This work has led to the advancement of novel drugs to clinical trials for Parkinson's disease. Current efforts focus on integrating genomic and stem cell technologies for targeted therapeutics. Dr. Khurana is a George C. Kotsias Fellow of the American Parkinson's Disease Association and an investigator of the Aligning Science across Parkinson's Initiative. He is a member of the board of directors of the MSA Coalition. He has co-founded 2 biotech companies that focus on CNS and neurodegenerative diseases. Along with Dr. Khurana, we also have other members who have joined us in putting together this webinar. One of them is here today. Ariana Pitaro is here joining us. And Ariana, would you like to introduce yourself briefly?
Yeah, thank you, Larry. Hello, everyone. My name is Ariana. I work at Brigham and Women's, actually with Dr. Khurana in the MSA clinic. And yeah, I'm just really excited to be here today. And I've been volunteering with the coalition for a little bit under a year now. So.
All right, thank you very much, Ariana. We have another member who couldn't make it today. Her name is Khurana Parator. Khurana has also helped us a great deal and has been a participating member in each of the webinars that we've presented so far. My name is Larry Kellerman. I'm also a member of the board of directors of the MSA Coalition. And I am glad to have the members who have joined us today. I want to thank Mariah Meeks, who is the one handling the technology. So Mariah, thank you very much. And now I think we'll move on and go to get to the actual presentation. The grant we will focus on today falls in the category of pathogenesis. For those of you who are unfamiliar with that term, pathogenesis means the process by which a disease or disorder develops. Dr. Khurana applied for a grant in our first year in 2013. It was titled Stem Cell-Based Therapeutics Platform for MSA. His research using those grant dollars earned him in June 2018 the Bishop Dr. Carl Golser Foundation Award's inaugural research prize for atypical Parkinsonisms. Today, we'll get to hear his story about the research. Welcome, Dr. Khurana, and thank you for taking the time to share today.
Yeah, you're welcome, Larry. It's great to be here as always. I'll give a brief talk. I have some slides. We don't necessarily need to get through all of these, but hopefully we'll cover some concepts. There'll then be a short break. If you don't mind, I'm going to need to relocate, but I'm going to then be able to answer any questions that might arise. Some of the work I'm going to present is it may be a little technical, but hopefully you'll get the gist of what I was sponsored to do, and we can answer questions as well. So hopefully you're seeing my screen now. Is that right?
Yes.
My screen. Okay. So I'm going to go into the full screen now. Thank you again for this opportunity. I'm going to put some of what I'm going to say into the context of, the big picture questions that we're trying to address at the coalition, especially through the collaborative cause. I've given excerpts of this before, so some of you may be familiar, but we don't really know what causes MSA. It's genetic and environmental factors. We don't know where it begins in the brain, in which cell. We don't have a model so far that is translated from a mouse into a successful clinical trial. We don't know what drives progression and how we measure it. And so we have inadequate biomarkers and biospecimen repositories, and we don't know how to really effectively treat it. We have symptomatic treatments. We certainly don't have any disease-modifying therapies. So this sounds pretty negative, really, as a way to start, but I actually feel the MSA community can and should benefit from tremendous advances that are happening for other diseases right now. We should not be reinventing the wheel. There are other rare diseases that have made big strides. ALS is a terrific example of a disease where they've kind of laid out a template for themselves to address these key questions. And this gives us a template in the MSA community to follow. And there are also more common diseases like Parkinson's disease, where similar challenges have been addressed over the last 20 years. And so we don't need to reinvent the wheel. But this inadequate level of information has led to the development of this concept of the collaborative core program for the MSA coalition, and it relates to these different cores that I mention here, to look at genetic and environmental factors, to create better models in the lab, and that's where my talk today comes in, to develop better biomarkers for the disease, core B, and to continue the seed fund program that Larry introduced to continue to push the envelope on seeding exciting ideas for treatments. So this is a challenge, and that's what we're trying to meet here. But it really comes down to the fact that MSA is a complicated disease. There's no clear genetic cause yet for MSA, and that differentiates us a lot from diseases like ALS or Parkinson's or Alzheimer's disease, where there's a clear gene. And I underscore that this is a real challenge, because it becomes very hard to model a disease in the lab where you don't understand the genetics of it. This is a limitation that we have. But there are things we know that can be helpful. So we think a one-size-fits-all strategy may not work because it's complicated, that we're going to have some problems with small-scale studies because we'll never know for sure if a particular toxicant in the environment or a particular genetic change is causing the disease. And we may need a human model for this disease. And so because this disease is really quite difficult, I've been, partly through the coalition's help through the seed fund, really developing, collecting stem cell models for some time. And I think now is the time to pull the trigger on a really systematic analysis of the impressive number of stem cell lines we have in the lab, because the technologies are there. But it is challenging. The reason why people have shied away from it is because, as I said, there's no gene in the dish that we can genetically correct to make sure that our stem cells are really telling us something meaningful about the disease. I'm going to talk to you about a protein called alpha-synuclein. There are patients with mutations in alpha-synuclein that have Parkinson's disease. We've been focusing on those stem cell models for a long time because we can genetically correct those mutations. So this has definitely been a barrier. That being said, there's been an explosion of stem cell technologies in the last, 5 to 10 years. And I think the time is now right to systematically develop a much better model of MSA, a human model in the lab. And so this is what we'll talk about today. Now, this is probably the most important slide because everyone's confused about what we mean by stem cells. We mean different things. And I just want to underscore what I mean today. I'll tell you first what I don't mean. I'm not talking about stem cells for transplantation. And that's not the work my lab does. And there are two types of transplantation. Firstly, you can replace the cell that's being lost. So many of you might have heard that dopamine neurons are being tested for transplantation for Parkinson's disease, certain retinal cells for retinal diseases. This likely doesn't work for a really complicated neurodegenerative disease like MSA because lots of different cells are being lost. I mean, it's possible that I'm completely wrong about this, but it wouldn't be the obvious choice for a disease where different circuits are being affected at the same time. And so that's a little different from Parkinson's disease. And then there's a different type of stem cell transplantation where you put in a cell that supports other brain cells. And for example, many of you who have been following the MSA trial field know that the Mayo Clinic is trying mesenchymal stem cells for transplantation. This is not to replace the cell that's already gone. It most likely doesn't bring back functions that are already lost, but the idea is that these cells secrete other factors that are helpful for the existing cells of the brain to thrive. So I want to be clear on that distinction. Now, the type of stem cell work we're talking about is quite different. It's using stem cells for drug discovery. So in this approach, a stem cell is made from a patient, converted into a cell type that's relevant for the patient's disease, and these cells can then be used for drug screening. So I hope that makes sense. But this can be a long process if you're going from screening a stem cell to a drug, and it's complicated, but it can be really useful, and this is where we'd love to use our stem cell models. It can be really useful if a drug already exists and you want to try and figure out which patient would this drug be best for. And that's really how we're thinking about our stem cell program right now. So this is the basis of this. These are called induced pluripotent stem cells. So here we take a skin biopsy from a patient, we make them into a skin cell, turn them back developmentally, the developmental clock back in time into these amazing cells that look like embryonic stem cells. They're called induced pluripotent stem cells. And from those, we can generate different types of cells that are lost in brain diseases. And as I'll review very quickly, what we're really interested in MSA are neurons and glial cells. These are heavily affected in this disease. So remember, MSA is a disease of alpha-synuclein aggregation, and this is somewhat fortunate because it shares this with Parkinson's disease and Lewy body disease. And what this means is a lot of companies that are developing therapies for alpha-synuclein, the protein that becomes toxic in this disease, they're doing it because there's a big market for Parkinson's disease, but they're really interested in trying these drugs also in MSA. So that the MSA field is benefiting from this biological relationship between the diseases. So just to remind you, synuclein exists in the cell. It then folds into the wrong shape. and then it causes these, what we call inclusions in brain cells. And in MSA, there are different cells affected. The glial cells, you may have heard of oligodendrocytes. These are characteristically affected by alpha-synuclein. This dark blob that you're seeing here is alpha-synuclein, and it shouldn't be there in that form. And it's now presumably disrupting these really important cells that provide the electrical cabling in the brain. These oligodendrocytes, the white matter cells, these are lost in MSA. And then the neurons are also heavily affected, and there's different types of alpha-synuclein inclusion that occurs in neurons. And so this is also very important. And so MSA, like Parkinson's, is a disease of synuclein, but it's in these characteristically in these glial cells as opposed to the neurons, although, as I just mentioned, MSA has very strong pathology in neurons as well, but it's not the defining feature. And it occurs in different distributions. So you will have heard of MSAP versus MSAC, and that really refers to where this pathology occurs, either at the back of the brain or in the middle of the brain. If it's at the back of the brain predominantly, it causes an ataxia, MSAC. If it's in the middle of the brain affecting the striatum and the dopamine neuron system, then you end up with something that can look a little bit like Parkinsonism, but because more of the circuit is damaged, it doesn't respond to dopamine. Now, alpha-synuclein turns out to fold into different shapes, and it turns out that these different shapes could be associated with these different synuclein diseases. And that's really important because the work that we've done really tries to capture, and what you see here in the bottom right here, is it tries to capture both the host, which means the actual cells from the patient, and the strains, these different alpha-synuclein forms. So it turns out that the form of alpha-synuclein that aggregates in MSA is different to Parkinson's disease, it's different to Lewy body disease. So as a first step in modeling MSA, we wanted to do something really simple. Rather than make the really complicated cells like these oligodendrocytes, we wanted to make simple neuronal cells quickly from patients and introduce the synuclein that's aggregating in that patient into the cell, and to do that quickly in the lab, so that if someone had a treatment, let's say for alpha-synuclein, an antibody, we might be able to test whether that antibody was good for that patient or not in this way. And so we wanted to develop this simple strategy to do it. And so based on the seed funding that I received in 2014, we've built a really, for a very, very rare disease like MSA, we've built a very solid collection of these iPSC lines from well-characterized patients, many of whom have matched postmortem brain. These patients have generously donated their brain, and so we hope we've built a resource for the community to better understand the disease. And I've been helped here a lot by Alain and Daesaba, whose photo you see here. He's an MD, PhD student. So now we have around 25 iPS lines or fibroblast lines from patients. 14 of those match to postmortem brains. And in total, we have around 50 synucleinopathy brains, in total around 20 MSA brains. So there's roughly 20 MSA brains, 25 iPS lines, and the intersection is about 14. So about 14 of those lines have a matched iPSC and brain, which is a great resource for the community. And this has been helped not only with this seed fund, but by additional funds over the years to create this resource. And so the idea here is that we can get matched stem cells and brain from patients at our center. Ariana was very modest in introducing herself, but she's doing a great job in building a clinical trial protocol for us at the Brigham. And here, every patient who gets into the trial ends up getting a stem cell made. If there are patients who pass away, they have donated their brain to us so that we can really understand the disease better. And I know having a question about, the question of brain donation is not an easy one, but it is very important for our research. So here you can see stem cells that have been made from a patient whose brain we have. So we have a matched stem cell line. In the top, you see that during this patient's life, we had biopsied the skin and had seen alpha-synuclein aggregating in the skin, but then we confirmed the diagnosis, and then we have the matched stem cell lines growing. You can see these embryonic stem cell-like cells. So this is how we want to advance really the understanding of the disease with this kind of collection. But for our patients who are, of course, wanting therapies, we need to have something that helps them in life. And that's exactly what we've tried to build from these stem cell models now, a system in which we can take synuclein from skin, spinal fluid, or brain. We can amplify it in the lab from a patient, and we can then introduce that back into their stem cells. And what we've done this now, we can do it quickly, and we can see alpha-synuclein forms aggregates that look a lot like what we see in the human brain, which is really great. It means we have a system where we might be able to test a synuclein therapy quite quickly in a patient, and we would love to kind of partner with our industry colleagues who have such treatments for the advantage of our patients. So this is basically how it looks. You can take an MSA patient 1, MSA patient 2. We can extract the alpha-synuclein, let's say from skin or spinal fluid. Spinal fluid's probably better, but we don't know enough. There is synuclein in the skin. We don't know enough about how they compare. And then we can introduce those into a brain cell model that we could make from that patient, or a kind of a standard cell line that we have engineered in the lab. And we can, and you can see that these are quite nicely matched to what we see in the postmortem brain. And then the idea is you could actually test a drug in this, and we're particularly interested in testing alpha-synuclein antibodies in this. And so we're talking to a couple of companies. This is a little outside their wheelhouse, but we're really trying to encourage them to be creative about their therapies so that we can all collectively learn how these therapies work and make them better. And so there's going to be new technologies now incorporated into Core M, when it gets funded. And so these were very simple neuronal models that I just showed that we've made, and we're submitting a paper to bioRxiv next week on this. But we, of course, now want to add those glial cells back in, really important other cells in the brain, so that we can start to see the complicated interactions that might happen when alpha-synuclein aggregates. This is a significant project. It's going to take us a couple of years to get off the ground, but we've got an amazing team with great technologies that can hopefully bring this together for the MSA community. We can even make mini brains in the dish that have not only, the neurons in them, but also the oligodendrocytes. And this would be work pioneered and spearheaded at the New York Stem Cell Foundation. And they're very keen to help the coalition do this, to create these better models for the disease. And we're even, if the coalition Core M gets funded as I hope it will, it involves a contribution by Steve Goldman at the University of Rochester, who's pioneered a technique in which he can make these glue cells, the glial cells that are so important in MSA, and he can make them from patients and actually introduce them into mice so that you have a living model with the human glial cells introduced into the mouse brain. So you can observe and analyze and study these cells over a long period of time. And we think this could be a very effective therapeutic kind of model to test potential drug therapies. So that's all I'm going to really say today. This has to be a collaborative effort, and that's why we have these collaborative cores. And I thank everyone who was involved in this, especially the coalition for starting the seed funds. And of course, we'll continue this journey together. Thank you so much.
Thank you, Dr. Khurana. I appreciate it very much. I know you said you have to take a short break to relocate and be back to answer any questions. We've got several questions that Ariana or I have drawn up. I don't see any by people who have been watching, but we'll see what happens as you're taking your break. You can go ahead if you need to. Is this a time you're needing to do it?
Yes, this is
All right.
perfect. I will take a little break now. Okay.
We will see you in 5.
Okay.
Thanks, Dr. Khurana. So there might be a couple of things to talk about while he is finding a new place to get seated so he can answer some questions. First, for those of you who are attending recently, the first weekend of October and the last day of September, we had our latest MSA Coalition National Conference. That conference was recorded and it has been available at the website where the conference was hosted. I believe it will soon be, if it's not already, at the MSA Coalition YouTube channel. So if you missed the conference or you saw a session and you would like to see it again, be prepared to go to the YouTube channel and you'll be able to find those sessions there. I think it might be also a good time, I'm going to wait till the end, but I would like to right now maybe let you know a bit about our last webinar. We've already sat down, Ariana and Khurana and I, and decided that we were going to keep the Wayback Machine going one more time and go another 2013 grant that was awarded to Dr. Roy Freeman. Dr. Roy Freeman asked for a grant. It's biomarker development in MSA. And Dr. Freeman is, let's see, he is at the Beth Israel Deaconess Medical Center in Boston, Massachusetts. He has been working with a number of doctors to see if he can find a biomarker by doing skin biopsies. And with that grant, he was able to get an NIH grant to help further the study. And so he will be bringing that information to us in December. I believe we haven't set the date yet, but I think it'll be the 2nd or 3rd Wednesday in December before Christmas that we'll be able to see that. So hopefully you'll be able to attend and bring others on as well. Ariana, I have a question for you if you don't mind. Are you available and ready to talk for a second?
I am available.
All right, so I know you're a student and you're a worker at his lab. Can you, or would you be willing to share by any chance some of what you do at your level of work? I know there might be somebody out there who has a daughter or a son who would like
Thank you
to
so
get in
much.
more detail.
So kind.
You back again, Vic?
Sorry.
Are you
Sorry,
back?
I'll be a couple of minutes, Larry.
Okay, well, we had a perfect thing to go to, so thanks. All right, so back to you, Ariana.
Yes.
Yes, would you like to share a little bit about sort of the work you do and how interesting it is, so that others can see what maybe is technically going on behind the research?
Sure. Yeah, no, I would love to. Thank you, Larry. So essentially, my role in the Khurana lab. So I work in the lab and also in the clinic. So it's kind of both sides of things. And essentially, the goal is to create this clinical trial paradigm at the Brigham. So we refer to it as N of few clinical trials. And the goal is to take small groups of patients with diseases such as MSA and really stratify them well and get to know their disease pathology well, get to know the symptoms that they have, collect biomarkers on them. We collect different biomarkers. We enroll them in studies that collect blood, that collect spinal fluid, that even collect stool samples, and we track them over time longitudinally. And we also do these brain scans. So we do specialized PET imaging, and we also do 3D MRI scans. And basically the goal is for us to track these patients every 6 months over the course of time. So that way, the better we know our patients, the more able we are to see if, let's say we introduce a therapy to them, does the therapy shift the curve, so to speak? If we see this disease progression happening, can we give a therapy and then see, does the progression kind of plateau, so to speak? So that's kind of the general idea of the work that I do in Dr. Khurana's lab. So really focusing on stratifying patients well to be clinical trial ready, we call it.
Very good. Thank you, Ariana. And by the way, Ariana volunteered long before we even thought we would have Dr. Khurana joining us. She was with us for the first and the second, and now this webinar as well. And she's been wanting to help the coalition make some sense of what's out there in the world today. And we appreciate that explanation, Ariana. Thank you very much.
Thank you, Larry. It's been a
I'm
pleasure.
back. I'm
Great.
I see Vic is back and ready, and he's got a window behind him, so he's ready to answer any
I'm ready.
questions that we have.
I'm ready. And yeah, Ariana's answer was so good that I feel like I really don't need to be here. She did an amazing job.
There you go,
That was
Ariana.
terrific.
Thank
Very
you.
good. Very good. Okay, we have two questions to start with, and then I think we've got a couple that we've generated ourselves to finish out anything that might be happening. We have one from Suzanne. Do you need to make a model for each patient? And then I have a second, maybe a follow-up from the same lady, from Suzanne. She also would like to know, have you tried any of the new antibodies, Lundbeck, Takeda? I'm sure you're not allowed to share, but just some questions, I guess. So those are two questions from the same patient.
These are really good questions. The answer is, so the question is, would we need to have first question was, do we need to have a personalized model from every patient? Well, in some ways, we hope not, because that would be obviously a major impediment. But we also would like to have a system that's scalable enough in case we do need to be able to do that kind of test. We wanted a system that was scalable enough to do that. And so I think at least in the order of tens of patients, I think our technology could really do it. It couldn't really do it for hundreds of patients, I think, but I think it can do it for scores of patients. I think we could have that kind of technology. The hope is we wouldn't need to do that. But one question is how many different synuclein strains are there in patients? And we don't know, but it's probably at least a few different types of strains in MSA and a few different types of synuclein forms in Parkinson's disease. And so it may be that we would get away with subtyping patients so that we wouldn't need to make a model for everyone. They would just be classified into hopefully one of three subtypes. Also, it might be nice if we could just screen the antibody against the synuclein from the patient, but we don't need to necessarily make a different stem cell from every patient. We could use the same stem cell and put synuclein from different patients into that same stem cell line. That would also make it a lot easier. So really, we don't know the answer to that. We've just built the technology now, and we're just starting to talk to companies about it. So we have not so far tested any active therapeutic against synuclein in our system, anything that's really in clinical trial. And really, this is because our technology is so new. Pharmaceutical companies are not used to doing this kind of work. They don't have this technology yet. So that's why we want to get our paper out, make sure that they understand what we've done. And I'm really hoping that once the paper's out, it'll solidify a number of conversations. But I will say that right now, we have two more mature conversations with companies that have antibodies that they want to test in MSA that have not hit clinical trial yet. And that's probably our sweet spot. Patients, a company that's already got a drug in a clinical trial is very protective of that drug. That's just a practical constraint that you all have to remember. If we were to use it and then find something about that we didn't like, that could jeopardize their program. So it ends up being a delicate conversation. And so our best bet is to take companies that haven't got into the clinic yet, but have therapies that they're really trying to get into the clinic that they want to, in their terms, de-risk. I think that's our sweet spot. And so we're talking to a few people right now. Can't guarantee that it'll work, but hoping that it will. And by the way, the therapies that we could test are not just synuclein antibodies. They could even be drugs like Vedipostat. You all may know that Vedipostat was a Biohaven drug. It didn't end up passing muster in clinical trial, but we've been actually investigating it in different patients, and we've made some stem cells from those patients, and we'd like to see if that drug has different effects in different patients. So we can use this technology in different ways. That's the answer, I think, to both of those questions.
Okay. We have another one from an anonymous attendee. Can we donate samples to your research or participate in any other way? So how might, maybe more generally, how do people enter the study and the clinical work that you do? Is there a possibility that it's open or not?
Yes, typically patients really, we're building a clinical trial ready cohort, as Ariana described it, from within our clinic. So certainly we can accept samples and sample donations. Most of the time, that would mean a visit to our clinic. Sometimes remotely, we can organize a skin biopsy, but that can be a little bit tricky. So remember, the way we do this is we take a small, tiny punch skin biopsy from the skin, and then we reprogram those cells. And so it can be done remotely, but really what we're set up to do is to do this for patients who we see in our clinic, so that that biopsy could be taken at the same time. But if there is a center near you that is willing to do a biopsy like this, we can certainly enroll patients remotely. That's just not been our standard practice. If patients are interested in being part of our clinical trial ready cohort, again, it typically is an in-person experience for us. We can do some things remotely, but typically it's really, instead of just seeing us for a clinic visit, we latch on the biospecimen collection to the clinic visit, and that's what Ariana organizes and orchestrates, and that's how we keep track of our patients. In the coming year, we're really hoping to add biometrics for our patients so that they can be remotely doing even apps on the computer, so we can monitor their movement longitudinally over time. The idea is to create a small cohort of very well characterized patients that could be ready for a clinical trial when one is available. And we hope that having this quantitative longitudinal data will end up, when we build a cohort like this, being really attractive to drug companies to partner with us, because we'd like to say, this is a complicated disease, but we have a patient's trajectory, we have their history, we know how they've been progressing, and we'll be able to tell you if your drug can shift the needle on how they've been progressing. And we just really hope that for those companies that are just about to think about a major clinical trial, that this kind of small, focused clinical trial will be very attractive, because they will learn something about their, about their drug and how to make it, how to design that definitive clinical trial. A lot of the drugs may be working in some patients, but if you don't design the right trial, you might lose the signal in the noise. And that's, we're trying to create a much more personalized medicine approach where, we're really tracking individual patients and understanding that. The last thing I'm going to say is that some therapeutics may come from within our department. We've worked in different ways on drug discovery, but for example, I have a senior colleague, Howard Weiner, right now, who's a very interesting drug that's being tested for MS, and he's interested in whether it might be useful for MSA as well. And Ariana has just helped me write a proposal. So if we can work with our colleagues internally and raise the funds to do this research, that might take out the complexity of interacting with pharmaceutical companies. And so we're exploring both of those avenues.
Okay. From that same individual has another question. I guess this could be more general answered as well. Can you test the drugs for excess iron with this model too?
We could. sorry, so drugs that are you talking, I assume they're thinking about the alterity type drugs that are out there that
That would
modulate
be
iron.
what I'm guessing, yes.
So yes, I think alterity has a theory that iron accumulation may be involved in different processes, including alpha-synuclein aggregation. I think, you know, we would need to partner with a company like that and see if our models really capture the right biology that they think is really important. A lot of their work has been done in mouse models, so that would need to be a conversation. But if there's an interaction between any pathway and alpha-synuclein aggregation, and if that pathway, that change in the cell, in other words, is engaged in our model, then of course we could test a drug in the dish like that. Yes, we could. I think you've just got to be aware of one of the limitations of our model. Right now, it's a rapid model that creates synuclein aggregates, but it's not a model in which the cells are aged. And that's something, you know, our collaborators actually here in New York, where I am, are working on very much, is how to make a more aged stem cell in the dish. And some of those things like iron accumulation could be really important as you age the brain cells. And so we may not see some of those key changes in the immature system, which is partly why we want to put the stem cells, human stem cells, into the mouse so we can study these in a longer period of time, because there's going to be processes that we can't capture in the dish.
Okay. I have one for you. There's no others up right now, but I have one for you. We now have the original population of those impacted by MSA were 40 to 65-year-old people. More recently, we found there's young onset and now late onset MSA as well. In your clinic, have you got a wide enough range that you can say whether the MSA looks similar for a younger patient as it does for an older patient? Do you see differences at all within the strains as you look, or is that something you've not had a chance to look at yet?
That's a really interesting question, Larry. We've had very scattered patients outside those age ranges, I would say, so probably not to hang our hat on, but you're right. I think, you know, the Parkinson's field is leading the way. The Fox Foundation is about to put out a study where they've really done a very systematic look, I think, at alpha-synuclein strains across a large population. So my sense from that data is that it's going to reveal that there are real differences between synuclein strains and different subtypes of patients. That could easily be the case in MSA. I think the problem is going to be numbers. This is a really tough thing in MSA, right? There's not enough numbers for us to make definitive conclusions about these things. We end up kind of using the Parkinson's data and extrapolating. I think that's a major issue.
I think you brought up in your message the idea that the brain donation is a very personal decision to make, but it is that and the sharing of the biospecimens as you're talking. Those are so important when you're dealing with such a rare disease as MSA.
Yes, absolutely. We don't have a chance unless we build a biobank, a genome bank. You know, what we're trying to do in the cause is to seed these different directions, but these are going to each need to be funded and supported in a robust way over time, so that we have the numbers to make conclusions. This is a major limitation. And as I say, the ALS field has been a real inspiration here. As you know, there are a number of therapies now that are shifting the needle on that disease, and I think that that's really impressive.
Yeah. Ariana, you had a couple of questions that we wanted to ask as well. You want to share one or both of yours?
Yeah, sure. So, Dr. Khurana, you talked about these antibody therapies that, we might be trying. Are there any kind of Parkinson's therapies that have shown to work for PD that might be able to carry over to MSA? Have there been any kind of crossovers like that?
It's only late September that we had our first, hopefully definitive positive news about an Alzheimer's disease drug, the amyloid antibody. It was a small effect, but it looks to be highly significant. There is, apart from a very rare amyloidosis, there is, to my knowledge, not a single drug that alters the course of a protein folding disease, and Parkinson's disease is one of them. That being said, some of the secondary analysis data of antibody trials in Parkinson's has been promising, and those definitive trials are underway, and some of those agents are going to be tested in MSA patients. So we're going to know a little bit about this in parallel, but remember, we don't have a disease modifying drug for Parkinson's right now, but we have a lot of potential disease modifying drugs for Parkinson's, and the companies that have made them are really interested in testing them in MSA. So I think this is going to happen a little bit, kind of in parallel. That's all I can say. We don't have that magic drug even for Parkinson's yet.
Okay. I got one, Ariana. I'll take one of the two that I shared with you. I went back to the actual 2013 grant, and in the appendix of that, you wrote, I am committed to bringing to bear my training to develop a multidisciplinary clinical research program focused specifically on MSA. What brought you to make that commitment?
That's so interesting. I didn't remember writing that. I didn't remember writing that.
I did a little research there.
Good for you, Larry. Well, I'm glad that, wow, we did follow up on that. As a fellow, as a movement disorders fellow, I started seeing patients in ataxia and Parkinsonism, and the one disease that I saw in both clinics was MSA, and it was because there was this really unusual and odd kind of double phenotype of MSA where you could present with MSAC or MSAP. And so I started seeing these patients, and honestly, it was just such an area of unmet need, and I just felt that we could do so much better for these patients. I thought the biology was likely to be very difficult, but if we were to learn something about the biology, we'd learn something really important about the brain because of the way the glial cells are affected in MSA. It's a very striking involvement of glial cells. So that's why I think there's both scientifically a lot to learn, and there's just such an unmet need in the clinic that I think it just compelled me to do that.
Okay, good. All right. I thank you for that. Ariana, you want to ask that last one I sent, or do you want me to ask it?
I can ask it if you'd like.
Go ahead.
Okay. So you noted in the summary of the 2013 that this work may lead to a novel functional genomics and small molecule discovery platform for MSA. Is it safe to say that you can now say that this did lead to that?
Yeah, I mean, I think that this is a first generation of models that we are happy to now share with the community. I'm very excited that really we've finished this first paper. It was a huge amount of work over about 5 years, but it really is rewarding to see that we have a simple form of a model that can be used for MSA, especially MSA synuclein therapy screening. And I'm just very happy that we were able to achieve that. But we haven't really tapped into the complexity of all the lines that we have. We haven't tapped into the complexity of creating more complicated models with both neurons and glial cells. We just really kind of, it's just chapter one, really. It's the preface, I would say, of the book. I think we need, I think the technologies are there, the interest is there, and that's what Core M is all about. We need to now take the next step and make a much more sophisticated model from human stem cells, and I think we should go ahead and do it. But I'm very, very happy that yes, we did deliver the first model that can be used for this. No one has a model, but our model can be used for Parkinson's, Lewy body disease, MSA equally. And so, yes, step one is done, but a long way to go, I think.
I think one area that you had pointed out in your presentation is the environmental exposure piece. We really don't have any knowledge of what's going on that side either. Yeah.
Right, exactly. So we're doing work on environmental exposures in Parkinson's disease because we end up having a lot more data there. As you know, with Beata Ritz, we're now actually introducing all of those toxicants into human stem cells, dopamine neurons from patients. We'd love to be here in a few years' time doing that for MSA toxicants as well. And that's why I'm hoping that she can give us some leads on that. But it's going to be challenging with the numbers she has.
Yeah, very good. Well, we're ready to close. I think I don't see any other questions here. And so, Ariana, you have anything more you want to say?
I don't think so, Larry. I think I'm all set for now.
Well, we want to thank Dr. Khurana for taking the time to share his research. We want to thank each and every one of you who donated monies to the MSA Coalition in the past and helped fund these last 3 webinars that we've shared. And a reminder, the next one in December will be with Dr. Roy Freeman and his biomarker study from 2013 as well. We have this year finished our awarding of the grants, and that will be soon announced, but the seed grants this year came in and we had 7 wonderful grants that you'll soon learn much more about. And in a future webinar as well, you'll learn more about them too. So thank you everyone for attending. Thank you, Vic. Thank you, Ariana. Thank you, Mariah. Everyone have a good Wednesday, and we'll see you all in December. From
Thank
Bench
you,
to
Larry.
Bedside, the review of MSA research. Bye-bye, Vic. See ya.
Thanks, Larry.
Thanks,
Thanks, Ariana. Thank you.
Yep.
Bye-bye.
Bye-bye.